All right, welcome back. We are now looking at chapter 29, which was split up in 29A and 29B, development and genetics. Cool beans? All right.
All right, first one, ectoderm, mesoderm, endoderm, what do they become?
Okay. When you are, before we get any farther, see how small sperm are compared to a big old egg? Anybody know why?
The sperm just holds 23 chromosomes. The egg has to have everything else.
Has to have everything else, including a yolk sac full of food for the embryo. Just like a baby chicken has a yolk sac to feed it, because there's no umbilical cord going through the shell. Got it? Well, there's no umbilical cord here yet either. Okay? All right. Gotta feed them though. All right. Let's see. What are we looking for?
Ectoderm, mesoderm, endoderm.
Okay. The three layers that we are. All right. See the three layers here? It's usually in blue, red, and kind of yellow. Okay? And the reason is because that is the color of the stain that attaches to those types of tissues. Okay? And so the upper layer is the ectoderm, bottom layer is the endoderm, and the one in the middle is mesoderm. All right? Now the question is, what do they become? Well, at the moment, see how they're part of the amnion, and this is part of the yolk sac. All right? Well, this blue layer, let me see this. I thought I had a picture of, there we go. There it is. Okay. This blue layer on top becomes three things. Number one, your brain, your spinal cord, brain and spinal cord, all of your nerves, and all of your epidermis. Okay? Now, this bottom layer down here, the one around the yolk sac, becomes the lining of all of your hollow organs inside your mouth, your esophagus, your trachea, your intestines. Got it? All the way down to the rectum. And these layers in the middle become everything else. That's all your bones, your muscles, your cartilage, okay, your connective tissues. Cool beans? All right. What else?
Function of oxytocin.
Okay, oxytocin has two functions. Primary, increase uterine contractions to get the baby out. Second function is milk letdown. Oxytocin doesn't make the mammaries create milk. Who's that?
You have to remind me.
Prolactin.
Prolactin. I should know that.
Pro, for lactation. Got it? So the prolactin, for the whole time you're pregnant, is making the milk happen. But it's not letting the milk out until the baby is born. And isn't that smart? Oxytocin says, here comes the baby, and lunch is served. Got it? At the same time. So that's the two functions of oxytocin.
Cool. And where is it synthesized?
Yes. That's the next question I'm going to ask. Where is it made? In the hypothalamus. And then it's released from the posterior pituitary. Remember? The posterior doesn't make anything. It simply releases the two hormones made in the hypothalamus. Who's the other one besides oxytocin? Starts with an A.
Aldosterone?
Nope.
No.
And it ends in H.
ADH.
ADH, right. D in the middle. Got it? ADH and oxytocin are the two that are released from the back.
Where's aldosterone from? Or where does it come from?
Adrenal cortex.
Okay.
Okay? The cortex of the adrenal gland.
All right. Eye color, skin color are examples of blank.
Okay, that's going to be a genetics question. That'll be next.
ABO blood system is an example.
That's also genetics.
Okay. ABGAR test.
Oh,
I'm sorry,
ABGAR.
ABG.
Yeah, okay.
ABGAR test.
I think the ABGAR test might be in here. See the little baby growing up? Yeah. Okay. All right. Oh, I don't think I got a picture in here, but I need to find it. When we first, another name for a sperm back in the old days was a seed, the seed of man. Why? because they believed there was a little person in the head of a sperm. And there's actually a picture that somebody looking at a sperm under the first microscopes, it showed a little person like this inside the head of a sperm cell. Because they figured, that's it. We make the baby, and mom is just a place the baby grew up in. They had no clue that there was a sperm and an egg with half the chromosomes. Because this, we're talking 1700s or whatever. They didn't know what DNA and chromosomes and all were. Okay? But that just, when I see that, it reminds me of that one drawing. It's an old woodcut, I don't know where I put that. All right. See, here is your spike in HCG from the baby to make us release more estrogen and progesterone. Got it?
Would that be even more than the corpus luteum?
Yeah, because the corpus luteum did this, and then the placenta does that. Remember, the first couple of months and then the last. Yeah, absolutely. And then, of course, when the baby's born, it all goes down. But notice, I don't have to do this the whole time. I do it as a big spike to get things started. Okay? Yeah, I knew that picture was in here somewhere. All right. That's how we have a baby. Okay, Apgar. It's named after this lady. She was a pediatrician, anesthetist, okay? And she invented a quick 5-step test to see how easily and how quickly kids are recovering from anesthesia, okay? Because that's very important. You have to know what's going on with your patient when you're an anesthesiologist. You put them under. Your job is to get them back out. Got it? Well, then she realized, hey, we could use this for a newborn. Her colleagues changed her 5 concepts, muscle tone, reflex, skin color, to reflect the 5 letters of her name. That's not going to be on your test. But where she was talking about muscle tone, they call it activity. Got it? Okay. Let me give you the bottom line. Basically, the Apgar test is a very simple grading system. If you see what you want to see, 2 points. If you don't see it at all, 0 points. If you're somewhere in between, it's 1 point. So, what's 5 times 2?
10.
10. So, a child with a 9 or a 10 or even an 8 total score is in good shape. 7 and below, now we got problems. Okay? So, activity. Is the baby moving? Yes. Not moving? 0. That could be a problem. Maybe just some flexing. Pulse. Is it over 100 like it's supposed to be? Is it absent? That's a problem, right? Is it somewhere in between? Reflexes. Is it pulling away? Is it doing something? Nothing. Or you pull on their arm and their leg and all they do is grimace. See, there's something maybe wrong with their muscles or their nervous system. Does that make sense? Okay, and then their skin color. Are they the normal color or are they blue? That's not good. Or somewhere in between, like just the arms and legs are blue. And then respiration. Are you crying? Are you breathing? Are you not breathing? Or is it somewhere in between? Got it? So quickly, boom, 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 add it up. There it is. Got it? And that's why so many doctors want babies to be born in a hospital, because if the Apgar score is low, you got a NICU unit you can spirit that little baby away to. Got it? So they can intubate it or IV it or whatever it is that's needed for that baby to be viable. Got it? Okay. But that's the Apgar score.
All right. Next up, ingredients in mother's milk.
Okay. We'll see. Okay, here we go. Milk. I was thinking we had a better picture of that. Okay, mother's milk, perfect fat. Yeah, it has a different fat and to protein content that cow's milk has. Also, it's full of antibodies, enzymes. It's full of IgA to fight off bacteria. It's got complement interferon, all sorts of healthy things for the baby, okay? As opposed to getting something else. 10-4. So that's what they never recommend cow's milk for obvious reasons. But we also don't just give them water. Got it? You got to have some nutrients in there, and that's why the closest thing, if you can't breastfeed, is Similac. Okay, where at least they try to put in as many ingredients as they can in powdered form for the baby.
With that though, you'll get the right fat and protein ratio, but you wouldn't get the
exactly all the other chemicals.
Yeah.
Right. Yeah.
Right.
But it beats the baby dying of starvation. So
Sure.
that's why you have to have it. Because some women cannot lactate.
I think I've heard that and correct me if I'm wrong, isn't there a thing where if you didn't drink your mom's milk, you might be more prone to getting sick and stuff easier?
I keep hearing that. I mean, it's anecdotal. I don't think I've ever seen a study on it. But it does make sense if you think about it. If mom is giving you antibodies to fight bacteria, well, you're too young to make your own antibodies yet. You know, your B cells haven't matured yet, so they can become activated. And they can't make antibodies when you're still a little tiny baby. Mm-hmm. So if mom can help you out, that's a big positive. So yes, maybe you get sicker. Now, there's something else, more of a conspiracy theory than anything else, so don't take it with a grain of salt. But we're getting sicker in this country year after year. Why? We don't know, because no one's looked at it. But it might be in the air, it might be in the water, it might be in the food that we consume with all the additives, preservatives, and fake sucralose or whatever else that's being put into our food. But we've never had this much autism before. We've never had juvenile diabetes like this before. You go back 60 years, that was almost unheard of. Now it's like 1 out of 24 kids. Where's this coming from? Same thing with autoimmune diseases. Got it? Mm-hmm. So we don't know. Is it lack of mother's milk or is it beyond that? 10-4? Okay.
All right.
Hopefully one of my students will do a study and figure this out in the future.
Okay. Timeline, pre-embryo, embryo, fetus.
Okay. Basically, if I have that picture, do I have that picture here? Nope, that's after the embryo. Well, all right. Okay. Basically, I'll make it simple for you. For the first 2 weeks, you are pre-embryo, okay? This could be anything. This could be a frog, this could be a person, this could be a dog.
And just to clarify, you said the first few weeks, this would be after the egg is
fertilized.
Yeah.
Mm-hmm. And we start dividing and making things, right? Okay. So, you're still you're pre-embryo for the first 2 weeks. Then you are an embryo. Now, an embryo looks like something. Got it? We can see a head kind of thing. We have some limb buds. We have a tail. Got it? But again, we call this an embryo because it's obviously a baby of some kind. But again, this could be a dog, a cat, a lizard. Okay? It's not until it starts looking like a person at about 9 weeks that we call it a fetus. Because now we can say, oh yeah, that's a human. That's not a dog, that's not a lizard, that's not a cat. Got it? So basically the timeline is first 2 weeks, pre-embryo, up until 9 weeks, it's a embryo, and then after the 9 weeks, it's a fetus. Because now it distinctly looks like what we're going to be when we grow up. Okay? You'll see the fingers and the toes and the sucking his thumb and all that kind of stuff already. Cool beans? That's as timeliney as it gets.
Cool. Let's see. Monospermy and polyspermy.
polyspermy. Okay.
Yeah.
Let's see if that's it here. Otherwise, I'll just tell you. Okay, where are you? Where are you, monospermy? Okay, here we go, monospermy. Okay, how many sperm do you want to make it to the egg? 1, monospermy. And we do it because the sperm are going to kick their way through all of this cellular material called the corona radiata, because it radiates out. Then it needs to use its acrosome to melt its way through this zona pellucida. And the sperm that is the first one to make contact with the membrane of the egg is the one that gets to put its DNA inside for fertilization. Make sense? Okay. However, just because there's one sperm over here doesn't mean there's not another one on this side. Got it? Oh, here. What's this? A polar body. Yeah. Off to the side. Okay. So, we want to ensure monospermy by blocking polyspermy. Got it? That's how that works. So, there are 2 ways to block it. Fast block, slow block. The fast block is as soon as the sperm makes contact with the membrane of the egg, the membrane depolarizes. So if it was positive on the outside, attracting a negative, it flips. Now it's negative. Will the negative attract the negative? That makes the other sperm push away because we flipped it. Got it? Just that quickly. Alrighty? That's the fast way. Okay? Prevent sperm from fusing with the oocyte or the egg membrane. Then there's a slower version called the slow block, which incidentally is called ZIP. And what this does, it destroys the receptors on any other sperm that might be attached and makes them detach. Got it? So they don't have a chance to come in. And that's how we make sure only one sperm gets in. We block the other sperm from getting in. Cool beans? All right.
All right. How to prevent?
What do we call more than one sperm getting in? Polyspermy.
Polyspermy.
So what do I call that genetically? Polyploid. Got it? Because what are we? Diploid. 2 sets of 23. If I got more than that, we're polyploid. What if I have 3 sets of 23? Triploid. Yeah. So, animals can't take that. People can't take that. But guess what organisms thrive on?
Plants.
Plants. If you've ever eaten one of those big giant strawberries that are dipped in chocolate, got it? That is big because it's octoploid. It has 8 sets of chromosomes. So plants actually thrive. They make bigger flowers, bigger fruits. You know, it's crazy. So they can actually survive with multiple sets of chromosomes.
Is that why when I can't remember what the test is, but when it was Darwin that took the flowers and you combine 2 types and it'll combine the colors to make a third color?
Yeah.
Is that...
No, that's not necessarily that. That is called incomplete dominance.
Okay.
Yeah, which we'll talk about in the genetics chapter in part B. Yeah.
All right. Let's see. Function of chorion, amnion, yolk sac.
Okay. The 4 sacs
that
are associated with a baby. Okay. That was just a little thing on in vitro fertilization that is in the news every once in a while. Okay. So, here we go. So, the chorion is the sac around the whole situation. Okay? In fact, let me see if there's a picture. Yeah. Okay. Ready? There's the chorion around everything. This is the amnion, which is going to be around the baby eventually. This is the yolk sac. Okay? And then sometimes you'll see on a plastic, see how this bulges out on the yolk sac? That's the allantoids, or allantois in French. Got it? This will eventually elongate and become the umbilical cord. Got it? Again, this is going to be the amnion that the baby floats in for protection and to have a homeostatic temperature the whole time. And then this is the yolk sac, which feeds the baby. Okay? See? There's the yolk sac. And as the umbilical cord grows, the yolk sac gets smaller and smaller. Because now we're going to start getting nutrients straight from mom's blood. We've used up the yolk. Does that make sense? But those are the 4 bags that we need to know. The chorion, we grow villi into mom's endometrium. Mom grows back, and that's the placenta. It's half junior and half mom. Okay? And then we have the amnion with the fluid where the baby is. And why do I want the baby floating at all times?
So they don't.
Floating in the fluid. Got it? Oops, that's all the floating pictures we have? Okay. Floating in the fluid. Why? Because we don't want a decubitus ulcer. We don't want a bed sore. It's the same reason we got to flip the little old lady at the nursing home. Because if you stay pressed down long enough, you're going to block blood flow and destroy those cells. We call that a decubitus ulcer. And the same thing is true with your brain. Your brain is floating because if it was laying down, the bottom of your brain would be crushed against the bottom of your skull. But it's floating in the fluid. And the same thing with the baby. Since the baby is floating, it can't get a decubitus ulcer on its butt or on its head or on its feet because it's constantly floating and flipping around. Does that make sense? Okay.
Let's see. All mitochondria are blank.
They all come from mom. Why? Because it's in the egg. The only thing dad contributes is the 23 chromosomes. Mom has to have all the other resources. All the Golgi, rough ER, smooth ER, the yolk for the food, everything mom. That's why the egg is this big and the sperm is this little. On purpose. And why is that important? Because we can trace ancestry through mitochondrial DNA. The mitochondria, even though it's only an organelle, has its own DNA that it carries from generation to generation. And it always comes from mom. Because again, dad doesn't contribute that. So we can look at mitochondrial DNA and trace your lineage back through moms. Got it? Through mom, through grandma, great-grandma, etc. 10-4? Because that's who you got yours from, which is really, really cool. They've been able to trace back, lineages as far back as, I don't know how far they've gone with some of these things. Okay?
All right. Results of non-dysfunction.
Okay, that's what this next picture is going to be, but I think that's on the next set of PowerPoints. Is there anything else?
Yes, 2 more. What is epigenetic
Okay, that's the next one.
And how does a child end up with double recessive?
That's also the next one. So
Okay.
give me a second and I will pull up the next PowerPoint. Let me make sure. Yeah. Okay. Super duper. All right. So, genetics, see? That's how it's supposed to work. Horizontal and vertical ends up. All right. That's just a little funny thing. Okay, what are some of the things I need?
So the first one, going back up, eye color, skin color are examples of blank.
Okay. Oh, by the way, see? Remember we talked about independent assortment? And see how all the purples line up? So that's what the gametes look like. But what if I switch one of them? Look how the gametes are different. And I switch to one in the middle. Gametes are different. And switch to one at the end. The gametes are different. Got it? That adds a layer of diversity. So we have crossing over, We have independent assortment, or segregation, as they say, separation, which this happens in anaphase 1, and crossing over is in prophase 1. But of course, the majority of our differences come from the fact that it's two different people coming together. Okay, and that's why we can end up with 70 trillion combinations in, 46 sets of chromosomes, 23 and 23. Got it? All right. See, and even more if you add diversity with crossing over and random mutations and stuff. All right. Now, let's make sure we understand.
I saw something about the skin and eye, and I think.
Did I already pass over? Those examples of dominant. All right. This will help with the question about how can a person end up with a
Double recessive.
double recessive. Okay. Just very briefly, you have to have, the male has to be heterozygous, dominant and recessive, and the female has to be dominant and recessive. Because look what happens. Double dominant, heterozygous, heterozygous. All three of these, because there's a capital A involved, only does what the capital A says. Got it? It's not until we have a recessive and a recessive coming together, so the chance is 25% of your children, 1 out of 4, will have a double recessive. Now, if that recessive is cystic fibrosis, or that recessive is sickle cell anemia, that's the child that gets it full blown. These two are carriers, like mom and dad were, because they're carrying the recessive trait. And this is double dominant, so they're normal. They don't have a recessive trait at all to display or to pass on. Got it? But that's, all three of these display a normal situation because they have the capital A. This one cannot display the normal. It has to display the disease. Does that make sense? Okay. And that's where it comes from. Having a carrier for a parent, both sides. They both have to be carriers or heterozygous. Hetero meaning different. They got two different versions. Okay? All right. The skin. You got to get the skin. Here we go. Skin and eye color. Got it? We call this polygenic. Poly, many. Many genes, one trait. It takes 3 genes to give you your skin color, 4 genes to give you your height, 3 genes for your eye color. How do we know this? Because is everybody's eye color on planet Earth either brown or blue? No. There's shades of brown and green in between. Is everybody 7 feet tall or 4 feet tall? No. There's shades in between. That proves that it takes more than 1 gene to make it. Okay? I don't know if I got a picture. Yeah. They did this. They took workers at their plant and had them line up by height. Okay? So, shortest on one end, tallest on the other end. Where do we have the majority? Somewhere in the middle. Which proves that height is not one or the other. Got it? You see, 5 fingers or 6 fingers, one or the other. Got it? Attached earlobe or free earlobe, it's one or the other. Roll your tongue or can't roll your tongue, it's one or the other. There is no halfway, which means it's 1 gene. If it's dominant, you can roll. If it's not dominant, you can't. 10-4? But there's not just 2 rows here. There's lots of rows. What they're saying is, look, when it comes to color, you can be very much light skin or very much dark skin. Those are the 2 extremes. Everybody else in the world is in the middle. Okay? Because it's a combination of 3 genes. Now, this is a great picture I get from the Biology 100 book. It's an old book we used to use, but I still like the picture. There's 3 locations that these genes are in. If all 6 are double recessive, that's blonde hair, blue eyes, light skin. Got it? If 1 is dominant, more darker skin, brown hair, maybe green eyes. A little more dominant, few more dominant, very dominant. Everything's dominant. Are you with me? And that's how we know it's more than one gene, because of all the variations in between. Got it? Okay.
Let's see. And then next is ABO blood system
Okay.
is example.
ABO blood type. Oh, the opposite of polygenic is pleiotropy. This is one gene gives you a bunch of traits. Got it? And the trait that is usually used is Marfan syndrome. You're usually over 6 feet tall, very thin, and very long fingers. Can you wrap your fingers around your wrist like that? I can barely make mine come together, much less overlap. Take a look here. I did this on purpose. If I put my wrist right here, look how much longer this guy's fingers are than mine. That's one of the signs of a Marfan person. Their fingers are twice as long as usual. I had a student in one of my classes, no, my wife had in one of her classes in Sumter High School, and he had his hands around a can of Coke, and his fingers went around the can of Coke back to his wrist. Okay? Because I had to do a double take. I went, what? And what am I seeing here? My wife said, yeah, he's a Marfan syndrome person. I said, how do you know? She goes, well, he told us that on first day of school. So people were quit looking at him. Okay? But that's what it is. In fact, he looked just like this guy. It's amazing. They think that Abraham Lincoln had that because he was extra tall with extra long fingers also. Okay? All right, but that's pleiotropy. Now, we did colorblind. I thought there was another one. That's problems. I'll do that in a second. Okay. I'm surprised. Did I pass ABO blood types real quick here?
I did not see it.
Polygenic, incomplete dominance. Yeah, here it is, ABO. It's called codominance. The A doesn't cover the B doesn't cover the A. If you inherit A and B, you have both of them displayed on your red blood cells. And as I've said before, I'm a classic example. My dad's blood type was B, my mom's blood type was A, I'm AB. See, I inherited both of them because they're both dominant.
If you have two parents that are AB, you would just still be AB.
Yeah.
Right? Okay.
Right. Okay? Does that make sense?
Yeah.
Okay. So that's codominance. This is incomplete dominance. This is the red flower and the white flower makes a pink flower because red doesn't cover but is not red next to white. It's a blend. It's somewhere in between. 10-4? Okay. All right. Does that make sense?
Yeah.
So.
All right. Next up is, let's see, results of non-disjunction.
Okay. Let's go to the picture here. Let's draw this. Okay. Again, I will use green for the male, and I'll make it simple. I'm just going to have, instead of making 4 sperm, let's make 2 sperm, okay? So, sperm here, sperm here. And we'll use pink for the ladies. And don't worry about making polar bodies. We got 2 eggs here, okay? All right. Fair enough? Just to keep it simple. Okay. What's the genetics that makes a guy a guy?
XY.
XY. There you go. So, when we have to split that into our gametes, 1 sperm gets the X and 1 sperm gets the Y. Okay? What makes a girl a girl?
XX.
That's right. 2 X's. So when we split that up, 1 egg gets an X, 1 egg gets an X. Okay? All right. Let's put these 2 together. Let's put these 2 together. All right? What's that going to be?
A boy.
A boy. Because he's going to have mom's X and dad's Y. Got it? What's this one going to have?
Girl.
Okay, it's going to be a girl.
2X.
Because it's going to be mom's X and dad's X. Got it? So, my friends, test question. Who determines the sex of the child every time?
The guy.
The dad. Because only the dad has two versions to give. Mom only has one version. Okay? Did I say something in the video about Henry VIII killing his wives? Okay.
I haven't seen it yet.
Okay. Sorry. Yeah. Henry blamed his wives for not giving him a male heir. Well, in their religion at the time, you couldn't divorce somebody. So he trumped up charges about them being heretics or committing adultery and had them beheaded. Got the next wife. Okay. Oh, another girl, baby? Next wife. Oh, and on and on. Got it? So, everybody cool with that? All right. Now, non-disjunction, I'll just use this. Non-disjunction means, and I know it sounds weird, it's a double negative. This is junction together. What's this? Disjunction. What's non-disjunction?
Stays together.
You stay together. You didn't come apart. Okay? Now, unfortunately, I'm not picking on the ladies, but guys grow new sperm every day. You're born with all the eggs you're ever going to have. All you do is release them. But they age with you. And as eggs get older, they are more prone to non-disjunction. Okay? So, what might happen is, instead of separating, this egg might end up with two X's, and this one doesn't have any. So, what's going to happen in this baby? You're going to have two X's and dad's Y. And this is called Klinefelter syndrome. Okay? This is Klinefelter. What's going to happen to this baby?
1X.
Yeah, it's 1X and nothing. This is called Turner syndrome. Okay? Now, what about this situation? What do I get here?
3X's.
Yeah, triple X. You become Vin Diesel. Got it? That's a movie. Triple X. That's called the triple X situation. Got it? Everybody with me? Okay, so let's take a look at what those look like. Just to give you a three-dimensional look at it. See, that's where I would be drawing those things on them. Here's Klinefelter. Got it? It's either mom has two X's and the Y, or dad didn't have disjunction and with a Y. Now what do you end up with? Take a look. See, you got two number ones and two number twos until you get down to the sex chromosomes. Two X's and a little tiny Y. Okay? So what do you end up with? A person who is male but has lots of female characteristics. Narrow shoulders, but wider hips. Not much hair. Okay, female growth of hair. Breasts. Okay, this is smaller than normal too. 10-4? And that's why a lot of them are the ones that undergo the sex change operations. I'm already this close. Let me finish it off. Got it? Not what's happening in these days. That's something else. I just want to be a girl. That's something different. This is actually a genetic situation called Klinefelter's. Okay? Now, how about Turner? That's Turner. We got two of these and two of these and two of these. Uh-oh, what do we have? 1X and nothing. What do you end up with? Shorter than normal. Other things like some lymphedema and cubitus valgus. What does that mean? You can't straighten your arms all the way out. Got it? They say a lot because the fossa isn't deep enough. Okay? That's a lady. I went to the website. She's Dr. Catherine Ward. So does that affect your mental capacity much? No, she got a PhD in genetics. Okay? But she's shorter than normal. 10'4". Unfortunately, both of these are also infertile. Got it? They can't have kids. You might have seen this lady on TV shows. She has this. That's why she's shorter, but she's still an actress. Got it? Okay. Now, what if you have triple X? Guess what? Thus, triple X syndrome most often causes no unusual physical features. I got this directly from the triple X website. Okay? Triple X kids are obviously female. Got it? But how come they don't have a lot of problems? And the reason I went to this website is because this is where parents whose kids are identified as triple X will upload pictures and stories. Got it? My child did this, my child is this. Because they want to see, is there any kind of a, abnormality? Got it? They might be taller than average, have some menstrual irregularities, but it wasn't really bad stuff. Does that make sense? Okay. And here's the reason why they think there's nothing really bad going on. How many X's does a girl have in her cells? 2. But each X tells you the same thing. Be a girl. Do you need to be told that twice? No. So guess what? One of the X's is turned off. How do we know that? Because when you do a cheek swab, and we used to do these in school before they said, oh, you can't do that, you might give somebody COVID. You're swabbing your own cheeks on your own. Anyway, don't get me started. We could see this. If you're a girl, see that black dot? There's no black dot inside a guy's nucleus. The black dot, we found out, is the turned off X chromosome. That's how you can tell in 1 second whether the cell came from a boy or a girl. If it's got a dot, it's a girl. Got it? So the theory is, I'll bet the dot is 2 turned off X's in a triple X person. Got it? That's why there's not so many irregularities. Got it? Now, we call that turned off thing a Barr body, named after Dr. Murray Barr. But we think it got turned off because of the work of Mary Lyon, who was the one who realized it's the X chromosome. Murray Barr noticed, hey, there's a black dot in only female nucleuses. Mary Lyon realized that's the X chromosome. It's been turned off by a process called lyonization, named after her. Got it? Okay. Does it make sense? All right.
All right. What is epigenetic control?
Okay. Epigenetic. What's epi mean? On top of. Got it? Oh, by the way, here's Down syndrome. That was another one of these non-disjunctions. With Down syndrome, it's not X and Y, it's number 21. If mom's egg has 2 21s and dad's egg has 1 21, how many 21s does the baby have? 3. That's Down syndrome. 3 sets of number chromosome 21. Got it? But notice, you get the exact same look, even in the animal kingdom. Okay? They're twins. Obviously, fraternal, not identical. He ended up with 3 21s, she's got 2 21s. Does that make sense? But that's what that is. And the reason I put this chart here, which I got offline, is this shows the frequency based on the age of the mom. See how Down syndrome takes off when you're a mom that's 40 plus? And that's because your eggs age with you if you're female. Then we kind of know that because here in South Carolina, Strom Thurmond had his first kids with his wife when he was 70. Okay, she was 35. And who just had a baby at 84 with his little girlfriend? Robert De Niro. At 84, he had a baby with a 35-year-old or something, 30-something-year-old. So 10-4, because guys are making new sperm all the time, so there's less non-disjunction possible. Okay, all right. Thalidomide, yeah, okay. What was the other thing you wanted?
Epigenetic control.
Okay, epigenetic. All right. See, the first level of genetic expression is what we've always known: transcription and translation. Right? DNA to RNA, RNA makes the protein. Boom, done. But now we know there's a second level, makes us a little bit more special, and that is when we make RNA, we cut out pieces of the RNA and only use certain pieces to make the protein. That's your second level, okay? The third level is there's a methyl group or an acetyl group that can land on a chromosome and either turn it on or turn it off. Got it? And that's why methylation inactivates one of the female's X chromosomes and turns it into a bar body. Alrighty? So, this is obviously has opened a whole new realm of study. Can we turn off a gene that makes something bad by putting a methyl group on it? Can we turn on a gene that will help us by putting an acetyl group on it? And that's epigenetics, that there's something epi on top of the gene that will turn it on. Remember, what's a gene? A piece of a chromosome that codes for one protein. Okay? So it's this piece is a gene, and this piece is a gene. Does that make sense? So we can turn them off or turn them on with these epigenic markers. Okay? That's, like I said, the newest line of research. Was there anything else?
Last one.
Oh, last one.
How does a child end up with double recessive trait? I feel like
And
we already talked about that.
That was the, both parents have to be carriers. They both have to
Underscored.
be heterozygous. And you end up with a double recessive trait.
Yeah, that's it.
Cool. Happy to help.